Okay, awesome. Well, thank you guys for being here and thanks for that introduction. As it was just mentioned, I'm going to be talking to you guys a little bit about my research on the Burgess Shale, principally a lot of the work that I did during my PhD, which I just wrapped up in 2018 on the Burgess Shale, which incorporated both species descriptions, but really was more focused on looking at entire ecosystems, so paleo communities in general, but also how we link different paleo communities together within the Burgess Shale. And so taking a bit more of a wide holistic view of this locality, which, I'm going to extol the virtues of in a moment, but I'm pretty sure I don't need to extol it too long to a group like you guys. So yeah, I'm really happy to be able to present that to you. I want to say right off the bat too, before I forget, that this kind of work, and when you see the amount of data that's presented, it really represents work that's been done for well, at least something on the order of 15 years, 2 decades-ish in terms of the data collection. And so this work that I'm presenting is going to be a lot of which is going to be my own. Also incorporates work from my own PhD supervisor at the Royal Ontario Museum, Dr. Jean-Bernard Caron. It incorporates work from some of the PhD theses of some of the students right now, who I'll mention when I get to those parts. Previous PhD students like Lauren O'Brien, who've also done paleo community work. And sort of hopefully this talk will tie it all together to show you guys a little bit about how we might want to think about the ecosystems of the Burgess Shale with all the data that's available to us now. So typically when people think of the Burgess Shale, I think this is the kind of image that comes to mind. And so again, I'm going to go into a bit of a brief description here about why the Burgess Shale is so important and the background and history of the Burgess Shale. Really, that's just going to set us up for thinking about the Burgess Shale in a modern context. And so, of course, the Burgess Shale was first discovered in 1909 by Charles Doolittle Walcott, who you can see on the bottom left of the screen here. And so the Burgess Shale is about 506-ish, 508-ish million years old. So it's Middle Cambrian. And I think most people know of the Burgess Shale probably through having read Wonderful Life. And so that's a very common way to sort of, be introduced to the locality itself and its importance. And on the right of the screen, you can see just an image from one of Walcott's field notebooks. It really doesn't look all that different from the kind of field notes that we take nowadays. Our field notes are still filled with sketches. I'm sure every paleontologist field notebook looks somewhat similar, just a lot of crude drawings of fossils that you see. And below that is what I think has become a relatively famous quote from Walcott in a letter that I believe was addressed to a colleague of his at the University of Toronto. And what he wrote was, I had a few days collecting in the Stephen Formation, which is the formation in which the Burgess Shale is, in the vicinity of Field in September, and found some very interesting things. And I think, well, it's nice and succinct, but it's a nice quote. And I think the reason it's become so popular is because it presages, I think, about 100 years of research that's going to occur on these series of localities. But it's quite a funny understatement of the incredible value of what he found. And Walcott obviously recognized a great deal of that value. But I think it's a testament to the Burgess Shale that hundreds of years, or over 100 years later, rather, we're still returning to the Walcott quarry and we're still making new discoveries, which is going to be the next 35 minutes of this talk. And so the reason, of course, that the Burgess Shale is so important, why it's kind of like, a mecca for anyone who works on the Cambrian or the early Paleozoic in general, or just paleontologists, and fossil enthusiasts in general, is the exceptional quality of preservation that we see there. And that's a term that gets used quite a bit. And, the Burgess Shale is the only place where, you could argue exceptional preservation occurs in the Cambrian, let alone the entire fossil record. But I'm going to spend a little bit of time explaining kind of like the magnitude of how exceptional it is. And I think it took me quite a while to realize the full extent of that magnitude when I started my PhD. And so here are 4 fossils from the Burgess Shale. These are pretty classic animals. Well, I'd say at least 3 of them are very classic in terms of being figured in Wonderful Life and on websites. And they represent extremely different body plans. They represent very different phyla. So we have Canadia spinosa up here. We have Anomalocaris, which is, probably one of the flagship animals of the Burgess Shale. It's typically what people think about when they think of an early predator. It's made its way into cartoons and all other sorts of media and things. It's got quite an interesting history behind it too, in terms of, kind of architecting the body plan of this animal from various different pieces before we had fully articulated specimens. So that's not a story for today, but obviously quite a celebrated fossil. Hallucigenia here, which again, another flagship animal, another very interesting story. I think in a lot of ways, equally interesting in terms of the history of the fossil itself as Anomalocaris, but again, not the focus here. And then we have Nectocaris, which is a mollusk, which has a body plan that's a little bit convergent on what we see in modern day cephalopods. So again, really fantastic fossils here. I think you can tell, even without me going into some of the minutia, how exceptional these fossils are, bearing in mind that something like Hallucigenia is a couple centimeters across, and we're seeing incredibly fine details on this fossil. And so it would be, I think they're immediately arresting. And so you obviously want to go to a place like the Burgess Shale to see them purely for their aesthetic value. But all of those details allow us to glean a lot of scientific information that we normally wouldn't be able to. I think what came up yesterday during Bruce Archibald's talk on some of those odinates was, sometimes you get information loss with two-dimensional compression, or if structures are sort of folded on top of each other such that they obscure each other, it can make it difficult sometimes to tease apart information if you don't have all the angles that you would want on your fossil. But with Burgess Shale type preservation and with the fossils of these localities, we can see all sorts of fine details that allow us to sort of get into the really nitty-gritty of the evolutionary affinities of these animals, which is particularly relevant when we're talking about the Cambrian explosion, the establishment of early diversity, but also their ecological affinities. And so really when you have fossils like the Burgess Shale, those from the Burgess Shale in terms of quality, and you have a decent amount of quantity that show the different sort of angles of preservation, we can really reconstruct these animals as they would have been in life and kind of help bridge that 500 million year gulf in time from any modern animal that we might see in a modern ocean to these ancient ecosystems with animals that in some ways look quite alien, but do have familiar characteristics. These animations, by the way, are on the Royal Ontario Museum Burgess Shale website, which you should definitely check out if you've not checked it out before, but I'm sure many of you guys have found your way over there before. And so I've highlighted a couple more fossils here, some of which were in the last slide as well. And I want to point out a couple of things. So really a few concrete examples of what I mean when I talk about exceptional preservation. So most of these fossils are quite small. I forgot to put on scale bars here, but I'll explain it in a second. Fortunately, I'm pretty familiar with them. And so some of the features that we can see here are incredibly fine features that you would not normally expect to be preserved. So going beyond just like, soft tissues that are in excess of shells, for example, or teeth or bones or whatever, we actually see features like eyes, which are, probably some of the softest tissues, but we see them actually in fantastic relief sometimes. This is an early chordate called Metasprigina. This again is Nectocaris, that early mollusk, and we can see their eyes and quite a great deal of detail on them as well. I think when people think of the preservation of eyes for invertebrates, they tend to think of things like trilobite eyes, or when you have a nice arthropod with a sort of like multi-lens, kind of like, very beautiful geometric eyes. We have what might be called sort of simple camera type eyes in these animals as well. This is a comb jelly up in the top right. We can actually see like the remnants of their comb rows. And so if you've ever, been scuba diving or snorkeling or watched an ocean documentary, you know how fine that these cilia, which are organized into little combs that pulse in time, can be. And we do see that on Ctenophorus and the Cambrian, like Ctenorhabdotis here. The bottom right is actually a fossil I described a couple of years ago called Kootenai which I'll return to in a little bit. This whole fossil is about 1.5 to 2 centimeters long in total length, I think. And so you can see this tiny structure here is actually a medial antenna. And so that just gives you, I think more than anything, a sense for how fine the features can be on these fossils. And I'll have a couple of close-ups later. And then of course, ecologically relevant features like the sort of forward-facing predatory raptorial appendages of something like Anomalocaris, but even fine suspension-feeding appendages that you find on animals like Lyricystis here. This is an early echinoderm. You can see how fine the individual, almost hair-like structures on their arms are, which they would have used for filter feeding. These aren't even necessarily the most sort of shocking examples of preservation you get in the Burgess Shale or in Burgess Shale type preservation. We obviously have guts as well, and sometimes we have the actual gut contents actually preserved in situ. So in things like predatory preapulid worms, you can sometimes see the remnants of their last meal actually in place. We can find things like putative nervous tissue in some of these animals. It's been demonstrated multiple times for arthropods, primarily from some other sites, but it has been recently demonstrated for annelids as well. Aspects of the nervous system actually preserved inside of these incredibly small fossils. So again, like I said, these kind of features are interesting from an evolutionary perspective, but also an ecological one, in that we have to sort of generally, unless you have catch an animal right in the act of some behavior, we have to infer their life mode based on their morphology. And these kinds of features allow us to do that. But what it also lets us do from a broader ecological perspective is reconstruct animal communities with greater fidelity than you normally would be able to. So on the top here is kind of an artistic representation of what you might typically think of when you think of a Cambrian locality. Only the hardest parts are being preserved here. So things like brachiopod shells, things like mollusk shells, obviously as well, the exoskeletons of trilobites, which sometimes might even just be molts or fragments of a molt. We have things like the individual ossicles of echinoderms. So that's your typical kind of Cambrian fauna. If you were to just pick and choose a random Cambrian locality, the kind of animals you might find. So relatively lower in diversity than something like the Burgess Shale, and also a resultant narrower view of ecology. And so what you would get is a very benthic scavenger sort of dominated view of the Cambrian Ocean if you were to look only at sites like that. If you expand this out into looking at the things like the Burgess Shale, where we have soft tissue preservation with great fidelity, as I said, we have a much more complex view of early animal ecosystems. So we have things like Anomalocaris, nectonic top predators. We have benthic suspension feeders like sponges. So you might get sponge spicules in some of these types of localities. We can actually get sponges preserved, with all of their kind of more soft tissues preserved, and the spicules in situ as well. Gives us a better idea of what the actual sponges look like. Epibenthic carnivores, entirely soft-bodied animals like Pacaya here, this early chordate which you see. And so I think quite frequently, the emphasis when you think about Burgess Shale type preservation and the animals in them is on the individual evolutionary insights that those fossils provide for whatever their group is. So the arthropods, mollusks, etc. But I think a little bit less talked about less frequently are the sort of broader ecological ecosystem level sort of ramifications. And so when you see an artistic reconstruction like this, a very dramatic reconstruction of, that's not Anomalocaris itself. I think that might be Lagania in the middle, but, a large radiodont rushing toward the small fleeing arthropod. You have preapulids, predatory worms in their burrows here, sponges assembled, small suspension feeding problematica, hallucigenia down here. This is not a massive stretch of the imagination, in that the fossils very much look like this. And given what you're going to see, the density of the fossils we find in exceptional localities like those found in the Burgess Shale, it's not a huge step of artistic imagination to go from here to there. And so that's, my little primer on the Burgess Shale. So obviously exceptional fossils there. The Burgess Shale itself is located up in the Canadian Rockies. But again, I think this is probably the group I need to say that the least to. Exciting sites. We have other sites, though, of similar quality preservation. I would still argue, although I'm aware that I'm biased here, I would still argue that the Burgess Shale gives us our best view on balance into early sort of middle Cambrian ecosystems, but we have other sites of similar quality of preservation and similar sort of quantity in terms of the raw abundance of fossils found. So probably the best known of these localities is Zhenjiang in China. I forget, I think it might have been sort of formally announced in the 80s, but it's been some decades since Zhenjiang was first discovered. A lot of fantastic fossils have been described from there, including very robust community ecological analyses as well. It's a community that I think has quite a few bit more preapulids. So those are or at least scaledophorans, sort of predatory worms, dominate many of the parts of the Zhenjiang biota. We have the Emu Bay shale in Australia, which some of you are probably familiar with too. There's quite a great number of worms there as well. The preservation is a little bit different. It's a very trilobite dominated community, as I recall. Sirius Passet in Greenland is a little bit older. The style of preservation, again, is a little bit different. Some of the fossils look a little bit more ghost-like, but there are some very weird animals from here as well, and particularly if you're a fan of early arthropods or sort of links between arthropods and their cousins, the lobopodians. Sirius Passet is a fantastic site for that kind of material. There's the Weeks Formation in the US, and so there are many Cambrian localities in the US, obviously, as the Rockies extend down as well. I would say that something like the Weeks Formation, for example, doesn't have quite Burgess Shale type preservation, or it's not quite equivalent to the Burgess Shale or Zhenjiang in terms of the quality of preservation, but they do preserve soft tissues. They do preserve a very fascinating and unique biota themselves. And actually, I just saw this paper recently and sort of announcing the Sousse location from Morocco. Again, the preservational quality, I didn't see as quite so many completely soft tissue animals as we do in the Burgess or in Xinjiang. But again, I just want to sort of broaden the spatiotemporal scope here in terms of what we're thinking about when we think of Cambrian localities. And so when you see them all arrayed like this, and this is just a small subset, there are multiple other localities in China, for example. The Haiyan biota was just described or announced, which is an interesting new site, which has a high number of juvenile specimens, actually, very interesting. There's obviously the Sinsk biota in Russia, so this is not comprehensive. I just tried to grab a kind of geographically disparate sampling. And when you look at a map like this, you might be tempted to think, well, we've had knowledge of the Burgess Shale for over 100 years. People have been returning to it over and over. It's been poured over. There's huge collections at the Smithsonian. There's huge collections at the Royal Ontario Museum, smaller collections at other locations. We must have a pretty good handle on it. And so the great promise in terms of understanding Cambrian ecosystems in the future might reasonably be said to be more in these other newer described localities. But I would disagree with that, and for a couple of reasons. The first is that most of our community ecosystem level knowledge about the Burgess Shale comes from the Walcott Quarry still. So the first quarry opened by Walcott. With most of the Burgess Shale sites that have been discovered lacking in either diversity of fossils, raw quantity, or even just accessibility compared to things like Fossil Ridge, where there's a nice good exposure. And so multi-locality studies of the Burgess Shale have historically been mostly constrained to broad scale comparisons, like how does the Burgess Shale overall compare to Xinjiang overall? Very valuable questions to ask, but fairly coarse grained in terms of the comparisons that you can make at that level. And so on this map here, you can see kind of yellow circles denoting a number of other Burgess Shale type localities that are known of varying quality. Some of them have tons of very promising stuff. Mount Oderé, for example, is one. I worked on some material from Mount Oderé recently, and I was kind of, I guess, recently reminded then of the incredible quality of some of that material. Stanley Glacier is another one that has a lot of fantastic fossil material. But overall, compared to the Walcott Quarry, it's hard to find equivalents in those three categories I mentioned before: accessibility, quality, and abundance. But we do have some. There is the Raymond Quarry located directly above Walcott Quarry. There is the Tulip Beds on Mount Stephen, which those two sites I'm going to be coming back to in a little bit. But what I'm really focusing on for this middle part of the talk is going to be Marble Canyon. And so I say most excitingly with the caveat that I did my PhD on Marble Canyon, so I'm very, very biased here. But it is an exciting discovery. It's a little bit old now. So it was originally discovered in 2012 by a Royal Ontario Museum-led team, but there were international collaborators as well, just a year before I started my PhD, actually. It's located 40 kilometers southeast of the original Walcott Quarry. And so you can see this is a view of Marble Canyon here. That's sort of me in the center and Jean Bernard down there. And so again, another view of the quarry sort of off from the side. And an initial paper came out on Marble Canyon in 2014 in Nature Communications. This is one of the figures from that paper showing the incredible quality of preservation. Again, in most respects, I would say equal to Walcott Quarry in terms of like what we might expect to be there, the quality of preservation, the fineness of some of these tissues. Again, here's a nice close-up on an arthropod's eye. It's kind of an inset in an inset, but you can trust me, the eyes are preserved fantastically. Again, there's eyes here. There are worms that are essentially entirely soft-bodied and you wouldn't expect to have preserved at all, except under the most exceptional circumstances. And when it was first described, it got a lot of popular press as well. I think someone must have said the word motherlode at some point, and then every news agency used that word, and it's appropriate because, as you'll see shortly, the quantity and quality of the fossils is just insane. And I know actually, I was looking at the sort of history of your symposia online, and I saw that Bob Gaines came to give a keynote speech a couple of years ago. And I think he was talking about Marble Canyon, specifically some of these discoveries in Kootenay. And I'm sure Bob came at it from a bit more of a geological angle, and also from the angle of, we just recently discovered this site. We have really only a small fraction of information about what we might potentially find there. And so this part of the talk, hopefully you can sort of see it as a logical follow-up to Bob's talk. They recruited me pretty much the year afterwards. I spent the next 5 years working on Marble Canyon material. So I can give you a bit more of a view into what we found in the intervening years. So obviously this entailed a good deal of quarrying. So I think their first analyses were based on about 2 weeks spent up there, just like picking up as much as they could after the discovery. We've been back there for in total at least another month and a half, probably close to 2 months of just quarrying time. You can see us all there in 2014, clearing off some overburden to start with, always the start. This gives you an idea of the sort of density or the amount of fossils that were coming out. And so this part's not, I guess, strictly to the science, but the way Marble Canyon Quarry looks. So once this was all cleared, essentially this whole area was used as kind of like a platform for people to be excavating fossils. With jackhammers and rock saws, we'd be cutting down columns, and then people would go, swarm them like ants with geological hammers to do the fine splitting to find the actual fossils. This is me up here, and on this platform just above them, I would essentially be surrounded by fossils all day. You can see that's Sydnea right there. You can actually ID some of these fossils from distant, that's how good they are. That's the worm Owesia hemicordate. And there were so many fossils to be organized, packed, prepared, and put into these cans for eventual airlift and shipping back to the Royal Ontario Museum that, I only really got to split rocks that first time for like the first week or two. And then there was such a mass of fossils that came out of this quarry that I could do nothing except try to keep up with recording all the fossils. You can only see me in this photo, but I'd often have a team of 3 people behind me also sort of reorganizing things, making sure all the fossils were identified with their counterparts, clearing them off. Obviously, sometimes we're being rained on here, so the fossils don't always look in the quarry, packaging them. And the other thing that never gets sort of seen in these photos is that you can see the sort of hint of someone down here. But the vast majority of these fossils, we didn't bring back to the museum, obviously. Not all of them are sort of that quality, but they are identifiable. And something that's going to differentiate, I think, some of the studies I tell you guys about briefly from your typical paleo community analysis is that we collected all the data, including fine scale stratigraphic data for all of the not collected specimens. And the way that happened was as these high quality fossils were being shipped to me and I was organizing it, anyone who found anything, it could be terrible quality, something that you would normally just chuck down a hill, they would just yell out whatever the fossil ID was. So they might yell, I might be in the middle of recording some data, and I might hear someone yell like 3 Bradorids, 2 Polychaetes, 1 Sydnea. And I would have to flip to this other book here and keep sort of 2 co-occurring records of everything we found in Marble Canyon. And so later on, when I quote to you a bit of the numbers in terms of the fossils we collected over that time period, hopefully that remember this picture and remember that's what went into it. That's the only way we could assemble a data set so large. When I get to the acknowledgement slides too, there's going to be something a line like, all the many people who helped us in the field. I couldn't even list them all. The crew was quite large. So before I get though to a broad sort of overview of Marble Canyon as a paleo community, looking at the Marble Canyon biota, I wanted to talk about a couple of individual studies that would not have been possible without material from the Marble Canyon. And so this is a paper, the first of 2 papers I want to show you from 2 of Jean Bernard's current PhD students. This is a paper lead authored by Joe Moisiuk, who's at U of T right now. And this is a radiodont. And so the same broad group that includes Anomalocaris, these large arthropods, but quite different from Anomalocaris. It belongs to a group called the Herdids, which have these large kind of almost like head shield-like carapaces. I believe this is the one that they called the spaceship when they first discovered it because, well, I mean, it looks kind of vaguely spaceship-like. I guess depending on what kind of spaceships you enjoy reading about in sci-fi. And it also has these incredible claws. And so if you think about the claws of Anomalocaris, they're quite long. They have spines projecting down, whereas those on this animal called Cambrobrancus are quite a bit more stout. You can see the actual claw itself is here, but they have these long, thin spines or cones projecting downwards, and then projecting forward off of these cones are another series of spines. And so these spines themselves, these secondary projecting ones, are actually quite fine. And so when we reconstruct the whole animal together, there are more articulated specimens than what I'm showing here. But imagine putting this all together, you get a reconstruction that looks a little bit like what you see on the right. And so when you think about the orientation of the arms, those combs and those spines projecting forwards, especially around this kind of radiodont circular plated mouth, what you have is almost like a basket-like arrangement, which could have like sort of projected forward to close and sort of bring foodstuffs back towards the mouth. And I believe in this paper, they sort of speculated that based on the fineness and the stoutness, or the fineness of these spines, the stoutness of these arms, that they may have been useful for sort of gathering food in a sort of deposit feeding kind of manner. So unlike Anomalocaris, which we think might have been probably a more active hunter scavenger, this is a more of a fine basket-like sifting set of arms. The next fossil I wanted to talk about is called Fibula caris. And this is, it's been described by another one of Jean Bernard's student, Alejandro Izquierdo Lopez. And I think it's a nice counterpoint to Cambrobranchus in that Cambrobranchus is huge by Cambrian standards. It's quite large and robust, whereas Fibula caris is so small that you'd be forgiven for missing it entirely if you were quarrying. You can see that down here, the scale bar is just about 2 millimeters. And so the whole animal itself is, about a centimeter in total length or less. And it's quite an odd specimen as well. So there's a broader carapace, which you can see here, the faint outlines of, but you can also see incredibly fine features that, again, you may not see under normal circumstances, under normal preservational conditions. So for example, the eyes, you can see a pair of them, they're kind of stalked here. You can see traces of the gut. And you can also see the impressions of extremely faint legs. So tons of tiny little legs. It's a beautiful paper, so I'm envious of them in that respect, but I don't envy the amount of work Alejandro had to put in terms of counting all these legs and doing this line drawing. It's incredibly fine, detailed work. And also, it's got this weird structure which looks like a spine. But a spine not projecting forward, but recurving back over the body. And so I think in our notes, we called it like the clothespin animal or something. What I remember is this animal is incredibly tiny, and I made a record of it somewhere, and we got back to the museum. Jean Bernard wanted to see it, and he was like, where's that paperclip or clothespin animal? And I don't even recall having seen it. That's how much it got kind of sidelined in my brain compared to these much larger, more initially impressive looking animals. That gives you a sense for its scale. But yeah, so this clothespin-like morphology then is a little bit confusing in that it seems to recurve over the area where you'd expect the legs to extend down if it was to be a crawling animal. And it's a really fantastic paper. But long story short, essentially, they've reconstructed this as an animal that probably would have swam upside down, which I think is, quite it strikes you as maybe a little bit odd or unusual. But then when you think of things like modern day triops, things like horseshoe crabs, which do swim by turning themselves upside down and rhythmically pulsating their legs, it's not that big of a stretch. But what it does do is illustrate, again, how the very fine details you see in Burgess Shale type localities can get us to a place where we might even describe the inverted swimming lifestyle of one of these minuscule centimeter long arthropods. And the last little individual species story I want to highlight from Marble Canyon is the annelid Kootenai Scolex barbarensis, which I described back in 2018. And so this won't be, a talk about the evolution of annelids, but specifically, again, I wanted to highlight it because this is a new species that's actually going to come into play when we describe the community ecosystem dynamics of Marble Canyon. But again, also the type of preservation we see here. So this entire specimen is about 2 centimeters long in total length, I believe. It's been quite some time since I've looked at them. But you can see actually a zoom in on just these structures that poke off the sides of a polychaete called the parapodia. They're kind of like fleshy shoulders. And then the individual bristles can be distinguished on a fossil like this, which are, micrometers sized in terms of total width. So that's the level of preservation that we're dealing with. I have a few other photos, which I didn't include in this talk, using SEMs to actually get accurate counts of the number of bristles on each one of these bundles. And so these are the types of fossils we find at Marble Canyon. These are the types of fossils that, if you recall Bob's talk, or if you read that initial paper, you would have seen a teaser of. And we now, with an additional, I guess it's 7 years-ish now of research, are starting to continue to describe. And this is a fantastic life reconstruction of Kootenai Scolex by Danielle Dufault, who is at the Royal Ontario Museum as well. She's a fantastic paleo artist. Okay, so we know something now about the incredible types of fossils that we might find at the Marble Canyon specifically, how they compare with those from other localities like Walcott Quarry, the type of preservation that we're dealing with. But now let's take the sort of one step back and look at the community overall. So this is the sort of top of the Marble Canyon quarry here, and that's me with the rock saw and Sean Bernard with a jackhammer. And so the overlying Eldon Formation, of course, the Burgess Shale is part of the Stephen Formation. So the overlying geological formation contact point is actually right about here, or about here on this photo, if we're looking at sort of a side view of the quarry. And again, to a group like this, I don't need to extol the virtues of having good stratigraphic information. But what we were doing essentially was using the Eldon-Stephen Formation contact point as our reference so that we could record the sort of 10 centimeter-ish scale occurrences of all the different fossils that we found here. So obviously, as we go further and further down, we're going further and further back in time. And we're actually recording the occurrences of all these fossils to the centimeter level, but we bin them into 10 centimeter intervals to account for some uncertainty, obviously, as we're recording the occurrences. So each of these 10 centimeter intervals are what I'm going to loosely refer to as a bedding assemblage, even though, of course, 10 centimeters represents snapshots of multiple sort of burial submarine mudslide events. And also I want to mention for those who are a little more like, data and methods conscious that for most of the analyses I'm going to show you, we removed the undersampled bedding assemblage intervals. So we were making, I guess we might call it more fair apples to apples comparisons in terms of data quality. And so what we have then now is a large assemblage of fossils, about 17,000 fossils included in these analyses of Marble Canyon, with stratigraphic information down to the 10 centimeter-ish level to assess patterns of, well, the biodiversity of Marble Canyon overall, now that we've spent about 2 months quarrying it, but then also the sort of individual turnover patterns of different groups of animals and how that results in changes temporally in ecological structure as well. So here's a brief overview in 2 parts. So we have a mixture of stratigraphically widespread and stratigraphically quite narrow species. So these are the 10 most abundant groups. There's a decent diversity there. Arthropods obviously are the most diverse and abundant, so that's no surprise, very similar to other Cambrian Lagerstätte. If you look at let's just say the top 4 species, Lianchinella, which is a small arthropod, Haplophrenitis, which is a hyolith, which many of you guys are probably familiar with, Oesia, which is a hemichordate, and Paranopsis, which is another arthropod, relative of trilobites. We can see that across the quarry, and you can see the sort of pictorial representation of it on the left here, they're all quite far-ranging in their temporal extent at Marble Canyon. You find them in most sort of intervals. If you were to give me a random chunk of Marble Canyon rock and I was to split it, I would have a reasonably good sense of surety that in that block, I would find some Lianchinella. I would probably find some Haplophrenitis. We have the opposite pattern for things like Kootenai scolex, that worm I mentioned before. It's very abundant, far more so actually than several of the kind of like typical Burgess Shale arthropods like Sydnea, but its stratigraphic distribution is a lot patchier and it's quite a bit more restricted in some areas. So in these areas in particular is where we find the vast majority of all the specimens. We find essentially no Kootenai scolex, just a meter or 2 above. So again, we're already starting to see some of the rudiments of complex spatiotemporal dynamics within the Marble Canyon biota. As I said, arthropods, the most abundant and diverse group, and you can see how it's very simply broken down here. And I want to point out too that many of these taxa are abundant at Marble Canyon, but rare anywhere else. There are also species that are entirely endemic to Marble Canyon as far as we know so far. But I think, again, a good example of this phenomenon is Kootenai scolex. And so we have good record of polychaetes in the Burgess Shale, dating back to Walcott's time. And Simon Conway Morris in particular did quite some fantastic work, I believe in the 80s, a monograph describing the species that were present there. But since then, we hadn't really discovered any new species of annelid from the Burgess Shale. So Kootenai scolex was the first one in, I think, about 40 years since Simon had described a number in his monograph. And a part of the reason for that is we only find now, with the knowledge that Kootenai scolex exists, we've been able to find a few from Walcott Quarry, but the vast majority of all of them occur only at Marble Canyon. To the extent that in some of these levels, and so this is actually around the 405, so it's down about here where I was just saying Kootenai scolex is particularly abundant. We have slabs like this, which are maybe 20 centimeters in total length. I've zoomed into it a little bit, where the entire slab is covered in Kootenai scolex specimens. these absolutely minute, maybe 2 centimeter long soft tissue worms preserved in incredible detail and, just blanketing the whole specimen. To the extent that if we zoom in on just this area of rock here, we do have a fantastic specimen of this polychaete, but we also have these very tiny ones that are probably like, an order of magnitude-ish smaller than Kootenai scolex, which we hypothesized might represent juveniles. And so within this very brief, 30 centimeter-ish set of strata, we have a ton of these polychaetes and a bunch of their juveniles, all sometimes within the single bedding horizon. So I think, again, that speaks to the kind of spatial or temporal, rather, variability that we can glean from Marble Canyon with a very in-depth look and with the kind of data that we collected. Looking at this another way, if you just look at the major taxonomic groups, so this is the groups in total, not just of the 10 most abundant species and how they change over time. So again, going further down here towards the lower bedding assemblages, we see the upper levels are dominated by hyalids, which you can see here, and hemichordates, which you can see right here. Well, it's a paper I published back in 2016. But around, maybe 3.5 meters below the Eldon-Stephen Formation contact point, arthropods start to become more abundant, particularly that small arthropod Liang-Chanella, which I described before, as well as some of these larger predatory arthropods like Tocumia and like Yaonek, which were described by a previous PhD student of Jean Bernard's named Cedric Aria. And then if we flip the view here, and instead of looking at taxonomic groups and how they change throughout Marble Canyon, and just flip this to look at instead at ecological groups, we see that, again, these upper levels are dominated by suspension feeding animals, the Lophophoridates, the suspension feeding hemichordates. And then when we get to these sort of mid to lower levels of the quarry, they're dominated in this sort of like violet color by nectobenthic predatory animals. So arthropods like you see here and here. So we do know that even within this quarry, if we were to have time averaged it and just looked at the community overall and compared it to Walcott Quarry, as is often done, we would sort of have a very muddy view of the overall ecosystem dynamics. And then we would be comparing, just one time average assemblage to another. But with the type of data that's collected here, we can see that there are significant changes in biotic and ecological structure, even within the span of 4 meters of strata. But of course, Marble Canyon is just the most recently described major site within the Burgess Shale. So there's, of course, the Walcott Quarry. There are the tulip beds, which I alluded to earlier, which was worked on by a previous PhD student, Lorna O'Brien. There's the Raymond Quarry, which is located just above Walcott Quarry by about 20 meters. That was opened by Percy Raymond. And so data is available from these sites as well, and was sort of awaiting a more holistic analysis, which is essentially why I signed on to do my PhD. And so on the right here, we've got a sort of a look at the Stephen Formation and how all the Burgess Shale sites stack up there stratigraphically. And on the left, we have just a conventional sort of topographic map. And so the kind of data that we're dealing with now is about 16,000 specimens from Marble Canyon, which we think is one of the uppermost localities within the Burgess Shale, possibly the highest or the youngest discovered site based on its proximity to the overlying Eldon Formation. We have the Walcott Quarry, of course. This is data collected. Lots of people have worked on this. Simon Conway Morris worked on the paleoecology of this group back in the 70s and 80s, or the paleoecology of the site back in the 70s and 80s. But my supervisor, or my previous supervisor, Jean Bernard, re-looked at a bunch of new material throughout his PhD and collected a further 43,000 observations. The Raymond Quarry, which I just alluded to, just above the Walcott Quarry, I collected another 9,000 observations towards the end of my PhD. So we had another site sort of stratigraphically in between these two. And then, as I said, Lorna, during her PhD, collected another 8,000 from the Tulip Beds locality, which we think is the oldest of these 4 localities, relatively speaking. And so for this last component of the talk, what I'm going to do is just show you a couple of analyses that take this whole data set and sort of analyze it together. And what I'm going to do is not really get into too much, hopefully, of the individual details, and take more of a broad view of what this means in terms of the distribution of these different animals across this group. And as a result, how we are starting to see the mosaic-like structure of the Burgess Shale paleo community. So here's an ordination with all of these taxa. So I think in total, it's like something like 77,000 individual specimens and about 200-ish species. So if you're not very familiar with ordinations, I mean, people are to varying degrees. The most important things to remember here are all species names are located physically close to other species that have similar distributions. So for example, something like Kootenai scolex has a very similar distribution across localities and across individual strata as Yonick, for example. And the other thing to note here is all of the dots represent those individual bedding assemblages of 10 centimeters, which I mentioned before. And so we're not just time averaging entire localities and throwing them into this data set. We're talking about at the level of individual 10 centimeter intervals. The exception being the tulip beds, which are time averaged as a result of it essentially being collected from a large talus slope. I've been to the tulip beds. Opening a quarry there is not a logistically very feasible thing. They've opened a small quarry, but it's less of a typical quarrying maneuver and more of a cliffhanger style quarrying action. And so getting 20,000 fossils while being suspended from ropes and with rock saws in your hand is a little bit less logistically viable than it is for these other sites. And so broad strokes, before I get into individual taxa here, we can see that the localities are quite differentiated from each other in multivariate space. So essentially just throwing all the data in together, they are quite differentiated from each other. There's minimal overlap in terms of the localities. And we can see that each of these localities also has a number of sort of not endemic necessarily species, but species that highly typify them. So there are way more species included in the analysis than I've included here. If I had included them all, this would be impossible to read, but let's just look at a few of these species. So here's the first set I want to talk about. And so these species in black are ones that are highly diagnostic of a set of localities. So what that means is, for example, if you were to open a random piece of Burgess Shale rock, not knowing what locality it came from, and you found a bunch of Atoya prolifica, there's a high likelihood that what you're finding or where that rock came from was either the Raymond Quarry or the Walcott Quarry, based on their incredible quantity at those localities, and also their lack of abundance at any other. The same goes for Isoxes, which is this beautiful, probably swimming arthropod, very big, kind of charming front-facing eyes, a really cool carapace that has these kind of spindle-like spine projections on either side. Your typical Morella splendens here, which is, again, no shock that it's mostly found in the Walcott Quarry. In fact, there are buckets, I'm pretty sure in the ROM collections or maybe the Smithsonian collections, old sort of buckets that were used to transport fossils where they just say on the side something like hundreds of Morella. Like that's the quantity in which they can sometimes be collected. Micrometra up here is found in great quantity at the Raymond Quarry. Not so much, I mean, not at all, I think, almost at Marble Canyon and very rarely at other sites. It's probably one of the most beautiful brachiopods from the Burgess Shale. It's got this incredible surface texture that almost looks woven, where there's a set of cross-cutting striations, and they're often preserved with their feeding setae kind of projected outwards in feeding posture. It's a really fantastic fossil. And then there's Liang Chanella, which I've talked about a couple times here, one of the most abundant arthropods at Marble Canyon, and actually also incredibly abundant at Walcott Quarry as well. So even though these four sites do have quite a bit of difference in terms of their biodiversity, they are, of course, some species that are shared across multiple sites, and in fact, some species that are shared across all sites. But I think more interesting, perhaps, are these species which I've described here. So these are species where the names are color-coded to match the localities that they're an indicator species for. That is, they're very ubiquitous and quite unique to that site. So things like Kootenai scolex, which I mentioned before, which is described from Marble Canyon. We have maybe 3 from Walcott Quarry, but for all intents and purposes, it is a Marble Canyon species. Yauna kootenaii and Tecumia as well are, indicators for Marble Canyon. If you were to find a specimen of Tecumia, the overwhelming likelihood is that you're in the Marble Canyon area rather than Walcott Quarry, for example. But all the localities have specimens like that, not just Marble Canyon. So Spartobranchus and Burgessia are two good examples of that same phenomenon for Walcott Quarry. This hemichordate worm that builds tubes. Burgessia, this beautiful arthropod, has a sort of semi-transparent dorsal carapace. There were the same things going on for Raymond Quarry. So things like Herpetogaster, this very enigmatic tentaculate animal with foliose tentacles, the sponge Alonia. And then at tulip beds, well, it gets the name tulip beds for this animal that's kind of tulip shaped, colloquially even known as the tulip animal before it had a formal name assigned to it, Cyphozactum. And so each of these localities, despite all being part of the Burgess Shale, and in the grand scheme of things, perhaps not that far from each other, Marble Canyon and Walcott Quarry are quite a distance from each other, about 40 kilometers, but relatively speaking, all part of the same geological formation, all sort of wrapped in the sort of public consciousness as just being part of the Burgess Shale, have significant differences in biological composition from each other, as revealed by these analyses and by about like 15 years of hard quarrying. And so to underscore this even further, at Marble Canyon, there's almost an entire absence of sponges. And so if we were to do one of these very coarse grained, like just look at one locality versus another, completely summed together into a single time average assemblage, you can see at tulip beds, sponges make up they're the fourth most abundant group of animals. Third, actually, if you were to sort of change this unknown category, which includes the tulip animal. At Walcott Quarry, they're the second most abundant group of animals. And this is very typical for a Burgess Shale type like Lagerstätte. You find lots of sponges, usually in significant diversity as well as abundance. But at Marble Canyon, we find essentially none. We have 5 sponge specimens, but really they're so badly degraded, we could barely even identify them as sponges. And so not just by looking at like, oh, well, I found a specimen of Kootenai scolex, so it must be in Marble Canyon. Also by just looking at the whole ecosystem, something is very different at Marble Canyon compared to Walcott Quarry, let's just say. And so it's tempting to maybe think of things like, oh, maybe there's some competitive exclusion going on or some biotic driver, like maybe sponges just can't hack it at Marble Canyon based on like the number of grazing animals there are, for example. But it's important to remember that we don't actually have direct overlap of these 4 sites stratigraphically with each other. So actually getting to a biotic interaction style explanation for these major changes is not a place that I'm super comfortable going. And so I think a more logical, perhaps, and more conservative explanation for this phenomenon is to consider gradual changes in paleo environment that are taking place over both spatial and temporal scales that make these localities different from each other. Marble Canyon different from Raymond Quarry, from Walcott, from tulip beds, that make the area more hospitable to some groups than others. So just for one example, since I brought up sponges, there are a whole myriad of factors that make some localities in extant environments good for sponges or not so great. So things like high turbidity, which might interfere with their unique feeding mode that involves lots of small chambers funneling in through their bodies. There are factors such as differing sediment types, which can inhibit the successful metamorphosis or the successful growth rather of sponge larvae. And so at least our working hypothesis then is that these kind of gradually shifting environmental parameters for which we don't currently have a great proxy are what is helping to structure the Burgess Shale in this way as sort of a mosaic of different localities that have different faunas associated with them, not just one large overarching monolithic Burgess Shale. And so let me just end with a couple of summarizing points here. And the first, and I hope something that you guys feel is true after having listened to this talk, is that our understanding of animals during the Cambrian is far from complete. And so that, I think, is probably not exactly a controversial view based on how many papers are published on the Cambrian every year. We publish a ton, but we still need to know a ton. If we were to zoom in even closer, though, our understanding of even just the Burgess Shale is far from complete. And so despite having, people having worked on it for over 100 years, not necessarily consistently every year, but despite over 100 years of knowledge, the Burgess Shale, our understanding of the Burgess Shale is far from complete. New species are discovered every year, or at least described every year, which fundamentally change how we understand the early evolution of major groups. And I highlighted a few of those before: the radiodonts, arthropods in general, the polychaete I showed you. But for every group you can imagine, mollusks, hemichordates, which are a specialty of mine, fossils from Cambrian Lagerstätte and the Burgess Shale in particular are consistently changing and causing us to revise our hypotheses about early animal evolution. And similarly, when new localities are discovered, it's not just a well for us to make more of those kind of evolutionary insights. Those localities themselves, by existing, change the way we understand ecosystem level dynamics during, really the advent of the first complex animal communities in my estimation. If there was an Ediacaran person here, I'm sure they would argue with that, but that's an argument I'm happy to have and have quite often. And so finally, the note I want to leave you on is what we need then is more field work. And I say that partially because field work is fantastic. And I don't think I need to extol those virtues to this group, but it's been quite some time since I've been out there and I'm really missing it. We need more holistic data sets. So when these papers are published in the individual species all the way up to whole site descriptions, we need more integration of those data sets so that we can analyze them in a sort of comprehensive framework. And of course, we need more rigorous analyses to suit those problems. So with that, there are lots of people to acknowledge. Obviously, the Royal Ontario Museum is doing consistent, incredible work there, and I was very happy to be part of that team during my PhD. So Jean-Bernard, obviously, and Bob, both of whom were contributors to the paleoecology project I described in the latter half of this project. And then there are too many other people to mention, although the two people I will sort of highlight here as well are Danielle Dufault and Marianne Collins, who are two fantastic paleo artists who have done a lot of the work artistic reconstruction wise that I've shown you. And then of course, various sources that have funded my research in the past, I should acknowledge as well. And with that, I'd be happy to take any questions.